Hi everyone, my name is Callahan, and this is my Unit 4 Case Connection assignment. For this assignment, I did cases 1 and 4, and this is my presentation. So for the first patient, which is a newborn, ammonium, or NH3, is produced in tissues throughout the body as a result of metabolism of compounds that contain nitrogen. Amino acid metabolism is the predominant source for ammonia in the body, and metabolism of amino acids includes transamination as well as oxidative deamination. The two primary amino acids that are the non-toxic carriers for ammonia are glutamine as well as alanine, and those are in blood circulation. Clearance of excess ammonia is through the urea cycle that takes place in the hepatocytes, and that is in the mitochondria as well as the cytoplasm. And to enter the urea cycle, you need the free ammonia, which is made through oxidative deamination. So the first step of amino acid metabolism, again, is transamination, and this involves the enzymes aminotransferases, and they require the cofactor peroxidyl phosphate, which is vitamin B6, and those are just involved in moving the nitrogen between these compounds. So you move the nitrogen from the amino acid, in this case it would be alanine, to alpha-ketoglutarate, and that forms the alpha-keto acid, which is pyruvate, as well as glutamate. You also have aminotransferases AST. In that case this would be glutamine, but however in this case we're referring to ALT, and this would be, again, alanine. The second step is in oxidative deamination, so that's just shown more in detail here. The first step would be the glutamate dehydrogenase, converting glutamate to alpha-ketoglutarate. This is the same thing as alpha-ketoglutarate. You also have the reversible reaction of NAD forming NADH, and you're forming the ammonium ion. The next step would be glutamine synthetase, and that transforms glutamate into glutamine. This is important because the body can't metabolize ammonia in its pure form because it's toxic to the body. The next step is glutaminase. That's an enzyme that reconverts the glutamine that was formed from glutamine synthetase back into glutamate, and this forms the free ammonia, which then can go into the urea cycle. So again, this takes place in the liver. Nitrogen transport is connected to gluconeogenesis through pyruvate and alanine. So here we see that this is alanine, and that would form pyruvate. Pyruvate is then what can be used in gluconeogenesis. Again, during exercise or periods of fasting, the body runs out of fats as well as carbs to metabolize it for energy, so we break down amino acids. However, in doing so, we form ammonia, which the skeletal muscles can't break down. So in that case, we form the alanine cycle, which is vital to transport this toxic substance to the liver. So that just allows for transport of this throughout the body and to the liver, and this allows for exercising or fasting muscles to get energy. The patient's elevated orotic acid level is most likely due to a deficiency in the enzyme OTC. So that's shown here. This is orotic acid, this is OTC, and this is due to an excess in carbonyl phosphate. So here we see it being shunted out of the mitochondria into the cytosol, and it's forming orotic acid because this is elevated. So under normal conditions in the urea cycle, this should not be elevated and form an orotic acid. The least likely enzyme to be deficient is carbonyl phosphate synthetase 1. This is due to the fact that an enzyme deficiency, and that would not cause an elevated orotic acid level. So therefore, it's probably most like an OTC deficiency. Furthermore, citrulline, the patient's low citrulline level is indicative of an OTC deficiency. Here you have ornithine and carbonyl phosphate forming citrulline via OTC. So a deficiency in OTC would thereby form low levels of citrulline. A deficiency in OTC or CPS1 would cause overall elevated ammonia as well as urea levels. In this case, the amino acid arginine becomes essential in cases of OTC deficiency. So that's shown here. Citrulline is the precursor here. You can see that pathway for arginine. So in the case of OTC deficiency, arginine becomes essential under normal conditions. It's not essential, but due to the abnormally low levels, it then becomes essential. The urea cycle is responsible for the de novo synthesis of arginine. Moving on to case 4, given the patient's symptoms and lab findings, the deficiency of vitamin B12 is the most probable cause. Vitamin B12, otherwise known as cobalamin, is used for a cofactor in a lot of different metabolic pathways. This includes the reduction of homocysteine to methionine, as well as methylmalonic acid to succinyl-CoA. It's also needed for DNA synthesis, nervous system, as well as red blood cell formation. Common symptoms include fatigue, irregular heartbeat, shortness of breath, altered mental status. A deficiency of iron is not most likely the cause of the patient's symptoms, just because of the suspected diagnosis of megaloblastic anemia. That's most indicative of vitamin B12 or folate deficiency, not iron. However, there could also be low hematocrit levels in iron deficiency anemia. Alongside iron, a B6 deficiency is not the correct diagnosis. That's due to the symptoms not really aligning with the hallmark symptoms of vitamin B6 deficiency, which are glossy tongue, cracked lips, and skin rashes. So while some of them do align, most of them do not, and as well as same with iron, vitamin B6 deficiency does not cause megaloblastic anemia. It would most likely cause the normocytic anemia. Here are just some vitamin B12 symptoms which align with our patients, especially the nervous system symptoms, as well as some of the bone marrow things, as well as like the rapid heartbeat and things of that nature. Vitamin B12 is vital to the process of DNA synthesis, and it is involved in purine synthesis. Vitamin B12 deficiency impairs purine metabolism due to some of the pathways involved. Again, vitamin B12 is required for the reaction of homocysteine to methionine. During this reaction, 5-methyl tetrahydrofolate combines with homocysteine to form tetrahydrofolate and methionine. In case of deficiency, the 5-methyl tetrahydrofolate is trapped, and the enzymes are not able to function as a result of the deficiency in vitamin B12. This interrupts DNA synthesis as well as purine synthesis. Megaloblastic anemia is a blood condition that is characterized by abnormally large red blood cells. And it's caused by deficiency in vitamin B12 or folate. So in this case would be vitamin B12 for the patient. It's most likely due to either low or missing intrinsic factor, which is how vitamin B12 is absorbed, or through poor diet. Various lab testing would confirm the patient's diagnosis. So the CBC confirmed that she had low hematocrit levels, but to further this, we'd probably want to do a reticulocyte count, and that just shows how many newly formed red blood cells are present. It would indicate pernicious anemia if that count is low. You could also do a vitamin B12 level, intrinsic factor, antibody level, homocysteine and methylmalonic acid levels. The intrinsic factor as well as the homocysteine and methylmalonic acid levels would be abnormal, so the last two would be elevated. In individuals with vitamin B12 deficiency anemia, we also might want to rule out iron deficiency anemia, so we'd be doing the iron studies, so iron binding as well as serum. And lastly, we would want to rule out folate deficiency by doing a folic acid level. So here's just a picture of two tongues. One is before treatment for pernicious anemia and one is after. So you can see here's the glossy tongue that the patient has. And that's my case study. And then these are just the references.